Hi, I'm Mr. Kurth and I'm going to be talking to you today about DNA, cloning and its role in evolution. The first thing I wanted to do was to just recap on what the typical animal cell looks like. So, just drawn one up on the board for you. You've got the cell membrane around the outside of the cell. You've got the nucleus in the middle, which I'll come back to in a minute, that's the most important bit. And then you've got the cytoplasm where all the chemical reactions happen. Okay, so cell membrane, cytoplasm, nucleus. Now the nucleus is the most important bit of a cell. Inside it's got 23 pairs of chromosomes. 23 times 2 is 46, so you've got 46 chromosomes, okay, in every cell apart from gametes in a human. The chromosomes are made from a substance called DNA. That's what the substance is called. So chromosomes are made from DNA. And they've got little sections, each chromosome's got little sections, and they are called genes. Hi, next thing I'm going to do is show you some DNA. Well, I'm going to get it ready, and then later on I will we'll have a little look at the chromosomes. Okay, so all living things have got DNA within their nucleuses. Okay, now here it's got a kiwi fruit, it's living. So what I've done is I've just prepared a couple of pieces, and all I'm going to do is literally get the kiwi fruit, grind it up. Okay, I really need to grind that up. Grind it down as well as I can. I'm not going to do this too long so that it's not too boring for you. That's it. Now, it could do with being a bit more ground up, but it doesn't matter. What I'm going to do is stick that in there. So we've got our kiwi fruit in a boiling tube. Next thing we need to add is some salt water. I prepared that earlier. Let's get that in there. And a little bit of fairy liquid. Now this just gets inside the nucleuses. Let's get the goggles on just to be safe. So get there. I'm just going to hold my hand over that in a good shape. Right. Next stage is to just put it in some hot water, about 50, for about 5 minutes. So we'll come back to that later on. So we've got in there, we've got the kiwi fruit, we've got the salt water, the fairy liquid, and now it's gone into some hot water. We'll come back to that later. Hi. The kiwi fruit, the salt water, and the washing up liquid has been in the hot water now for about 5 minutes. So the next stage is to just add some cold ethanol, and this should reveal the chromosome strands for us. Okay, if we just do that, and then floating at the top, if you could just zoom in on that, this floating at the top, they are all chromosomes that have come from the inside of the nucleuses of those plant cells there. Right, next I'm going to talk to you about mutations within the nucleus and mutations to the DNA. Okay, so let's first of all make a cell. We've got the cell body there with the cell membrane and the nucleus, and then we've got some chromosomes here. Now these have only got 4 chromosomes there, but I'm sure you can remember in a regular human cell there are 46 chromosomes. I've only done 4 for now. They pair up, so there's 23 pairs or 46 in total. Now, those chromosomes are made from DNA. Well, what's DNA made from? DNA is made from bases, and I've set something up back here for you. Now, I've got one of the chromosomes from over there. The chromosomes are made from bases. Now, there's 246 million bases up one side, and then another 246 bases up the other side of the chromosome. So in one chromosome, 246 million. Another 246 million. Down, they're a little bit like a ladder. So you've got the order down there, and then the order down there. Now, they pair up. There's 4 different base pairs, C and G, A and T. So they're the 4 different ones, and they always pair up in this way. A pairs up with T, C with G. You can see further up here, A and T, C and G again. But sometimes they are the other way around as well. They don't necessarily order themselves in that way. So it's a little bit like that. It's got the bases like that, but hold whole sequence is twisted like that, a little bit like a twisted ladder. Okay, and they're joined across there like that. Now, what causes a change to this sequence is one of three things: either chemicals, radiation, or viruses. These are three things that can change this sequence here. So let's mutate this DNA. Some viruses, some radiation, maybe some chemicals, one of them. Okay? And what can happen is they can change around like this. We might get a full change like that. And then because we've changed the DNA sequence, the chromosomes change, but more specifically, the gene inside is changed. Just to finish with, I want to summarise everything we've talked about today. So, typical human cell. Every human cell has got a nucleus in it and 46 chromosomes. Those chromosomes are made from a substance called DNA, and they're arranged in sections called genes. Now, you've probably got about 20,000 to 25,000 genes within each nucleus. Okay, that's the best estimate we've got. Now, the chromosomes themselves are made from a substance called DNA, which is made from bases. Now, sometimes these bases change their order, and there's three things: chemicals, viruses and radiation. Those three things can change the order here. So, you know, you might get a mutation like that. And then if you do get a mutation like this, let's swap that one around as well. If you get a mutation like that, you get a whole new gene. And that is what can cause evolution and a change to a species. Hi, next thing I want to talk through is cloning. Okay, now the simplest way of doing that is to actually get a zygote, a fertilised egg cell, and take that fertilised egg cell and let it multiply. Okay, let it multiply until you've got a nice big ball of cells, an embryo. Okay, now the simplest way to do it is to just take that embryo from the uterus and separate it out. So you've got two lots of cells, and then let these multiply into new embryos, and then just take those and implant those into another uterus. You will then get a clone. This one would be a clone of this original cell. Okay. Another way of cloning is to actually get a cell from the embryo that you would like to clone, and remove the nucleus from it using chemicals, and get that, keep that nice and safe. What you can then do is get, for example, an empty egg cell, remove that from that. Okay, now what you've got is you've got an empty cell here, but that doesn't matter. This is the animal we're going to clone, and we've got the nucleus from there. Now all you need to do is give this empty egg cell and this nucleus a mild electric shock, and that will fuse, and you will get, it's called fusion cell cloning, and then this will develop into a clone of that original cell. Right, just to finish with today, I just want to summarise everything that we've talked through. So, it's a typical human cell. It's got a nucleus and it's got 46 chromosomes in it. Those chromosomes are made from DNA, and they've got sections called genes. Come over, I'll show you what's DNA made from? Well, it's made from bases. Now these bases come in pairs, C and G, A and T, G and C, and T and A. So these are the four base pairs, and they line up like that. Now, a couple of things can cause these genes to mutate. Chemicals, radiation and viruses could maybe change these around or change the order around, and then we get new genes and we can get evolutionary changes to species. Also, with regards to cloning, just in summary, cloning, what we can do is we can allow the cell to multiply. We can then, when we've got our embryo, our ball of cells, we can separate that out and take each new ball of cells, or each cell, let it develop, implant it, and we will get, from this, we could get two clones. Or, the second way of doing it is to remove a nucleus and maybe transfer a nucleus, give it a mild electric shock. This could develop into a clone of this. And all it is removing the nucleus from the animal you want to clone and putting it into a new empty egg cell, and then it will develop into a clone of your original organism.